Biology textbooks claim life is fragile, limited by temperature, water, and aging. But there are animals proven to shatter every one of those rules. Scientists have found tardigrades surviving extremes that should destroy all known life, jellyfish that reset their own age, and creatures thriving where logic says nothing should. If even our core biological laws can break, what else about life is waiting to be rewritten? Biology draws its boundaries with numbers. For most known life, the range is strict. Cells function only between freezing and scalding, but rarely outside 0 to 40 degrees Celsius. Slip below or above, and proteins twist out of shape, membranes break apart, or chemical reactions grind to a halt. Water is not just a helpful ingredient, it's the solvent that lets every molecule inside a cell move, react, and repair. Without it, enzymes seize, genetic material unravels, and metabolism collapses. Homeostasis is the name for this delicate balance, a constant battle to keep temperature and hydration in a narrow, life-sustaining band. Even a brief loss of water or a sharp temperature swing can mean irreversible damage. In textbooks and lab experiments, this is the rule. Every animal, from the tiniest insect to the largest mammal, relies on steady conditions to survive. These limits are not just suggestions, they are supposed to be the unbreakable laws of biology, defining where life can exist and where it cannot. A tardigrade can survive temperatures that would destroy the molecules in almost any other animal. In laboratory tests, some species revived after being chilled to -272 degrees Celsius, just a fraction above absolute zero. Others withstood dry heat at 150 degrees for a full minute. These creatures endure the loss of nearly all their body water, shrinking into a compact, glassy form called a ton, where metabolism falls silent and time almost stops. In this state, over 98% of water is gone, yet the cell structures remain intact thanks to a molecular toolkit that reads like science fiction. SAX-D and other heat-soluble proteins act as flexible shields, locking cell parts in place while a sugar called trehalose forms a glassy matrix. DNA repair proteins, including the now famous D-soup, stand guard, binding to chromosomes and absorbing the shock of radiation or chemical attack. In 2008, a European Space Agency mission sent tardigrades into open space. They floated in the vacuum, bombarded by cosmic rays, then returned to Earth. 68% revived, some even laying eggs. Dr. Ingemar Jonsson, who led the mission, described his disbelief as he watched the animals stir back to life in his lab. The full recipe for this resilience remains out of reach. Scientists can insert SERP into human cells and see some protection, but nothing close to what the whole animal achieves. The secret of the tun, how it preserves life at the edge of physics, still eludes us. A single jellyfish, smaller than a fingernail, drifts through the water, seemingly ordinary, yet it holds a power that biology says should be impossible. When threatened by starvation or injury, Turritopsis dohrnii does not wither or die. Instead, its mature medusa body collapses inward, forming a tiny, shapeless cyst. Inside, something extraordinary unfolds. Cells that once knew their purpose, muscle, nerve, stinging tentacle, shed their identities. Through a process called transdifferentiation, they revert to a youthful state, guided by a suite of genes that include amplified DNA repair enzymes and telomere maintenance programs. Telomerase activity, usually reserved for early development in other animals, persists here, protecting the genome from the clockwork of aging. In laboratory colonies, some individuals have cycled through this reversal for over a decade, never succumbing to old age. Japanese researcher Shin Kubota has devoted years to tending these colonies, recording each transformation with a sense of awe and unease. He once speculated that his jellyfish might outlive him, as the creatures returned again and again to their earliest form. Yet, for all the data collected, the true trigger for this reset remains hidden. Scientists debate whether immortality is the right word, or if these jellyfish are simply masters at escaping death's approach. The mechanism, distinct from mere survival, hints at a loophole in the rules of life, a secret that, for now, belongs only to the jellyfish. A severed limb is not a death sentence for an axolotl. Where most animals heal with scars, the axolotl launches a different response. Cells at the wound's edge shed their specialties, returning to a flexible, embryonic state. This mass, called a blastema, is the starting point for something biology textbooks say shouldn't happen after birth: a complete rebuild. Molecular signals take over. FGF8 surges, coaxing cells to multiply, while the WNT pathway quietly mutes the usual scarring response, clearing the way for new tissue. Over weeks, the limb regrows, bone and muscle reappearing in perfect order, nerves weaving through fresh skin. This ability comes from neoteny, the axolotl's refusal to grow up. It keeps traits from its larval stage, including the power to regenerate, not just limbs, but parts of the heart, spinal cord, and even the brain. Dr. Emma Bryson once compared the process to a living Lego set, where each block knows when to rebuild and where to fit. Yet, when scientists tried to transplant these pathways into mammal cells, using CRISPR to boost FGF8 and tweak WNT, results fell short. Mouse tissues healed faster, but blastemas never formed. Scar tissue always took over. The axolotl's secret remains locked in its unique biology, suggesting that the rules of development are not as rigid as once believed, but the blueprint for true regeneration is still out of reach. In the icy waters off Antarctica, a family of fish swims with blood as clear as glass. Unlike every other vertebrate, these ice fish have lost hemoglobin entirely, the molecule most animals depend on to carry oxygen. Standard biology insists that without hemoglobin, tissues would suffocate, especially in sub-zero seas. Yet ice fish thrive at temperatures down to -1.9 degrees Celsius, where water itself should freeze solid. Their secret lies in a radical redesign of the vertebrate body. Antifreeze glycoproteins, unique to these fish, circulate in the blood and tissues, binding to tiny ice crystals and halting their growth before they can cause fatal damage. But without hemoglobin, oxygen dissolves directly into their plasma, a process far less efficient than the red blood cells of other fish. To compensate, ice fish have evolved massive hearts that pump 3 times more blood per minute, and gills so large they almost seem out of scale. Genetic analysis by Dr. Mark O'Neill and his team revealed the loss of the entire alpha-globin gene cluster about 20 million years ago. A deletion so rare, it upends the idea that some molecules are universal. As Dr. Neer described it, the engine has been rewired. The icefish's clear blood is a living contradiction, proof that even the most basic rules of biology can be rewritten by evolution's hand. A mile beneath the ocean's surface, where sunlight never reaches, the rules of life change completely. In 1977, the submersible Alvin descended to the Galapagos Rift and found something that should not have existed: entire ecosystems thriving in total darkness. Instead of drawing energy from the sun, these communities survive on chemical reactions alone. At the heart of each hydrothermal vent, plumes of hydrogen sulfide billow into the water, poison to most known creatures. Yet here, dense clusters of tube worms, clams and shrimp gather, nourished by bacteria that perform chemosynthesis. These bacteria, equipped with the SOX gene pathway, strip electrons from sulfur molecules, building organic matter from carbon dioxide without a single photon. Under pressures reaching 1,000 times what we feel at sea level, their cell membranes remain fluid, thanks to unique lipid structures, unsaturated fats that resist compression, allowing life to function where proteins should collapse. Dr. Maria Ortiz, a marine biologist, compares these vent oases to coral reefs, but with a twist. Every gram of life here is built on chemical energy, not sunlight. Genomic studies reveal that vent bacteria adapt their membranes and enzymes to survive both the crushing pressure and the toxic chemistry. The discovery of these sunless worlds forced biologists to rethink the very definition of habitability. If life can flourish in such alien conditions, the boundaries of biology may be wider than anyone imagined. Biology's outliers aren't just curiosities, they redefine what life can endure, as researchers race to harness tardigrade genes or unlock axolotl regeneration. The boundaries of medicine, aging, and even space exploration shift. Nature's rule breakers remind us life's possibilities are wilder and wider than we've dared to imagine. What would you change if biology had no limits?